What if I told you there's a machine that can actually teach your brain how to smell? And I don't just mean detecting a scent, I mean pinpointing its exact source with almost superhuman accuracy. It sounds like something straight out of science fiction, right? Well, it's very real, and we're gonna break down how it works.
So when we say a superhuman sense of smell, what are we really talking about? It's way more than just knowing a chemical is in the air. That's the easy part. Think about a bloodhound on a trail. It doesn't just go, yep, I smell it. No, it knows exactly which direction to go to find what it's looking for.
And that is the real game changer here. The hard part isn't just detection, it's what's called ranging, the ability to create a vector, basically an arrow in the air that points you directly to the source of that smell. And honestly, our best technology today, it can't even come close to doing that. But here's the wild part. A solution actually exists. It's a biohybrid system called ODAR. That stands for olfactory detection and ranging. But the entire network, and I mean every single sensor out in the field doing the work, is completely dependent on one single, extraordinary machine sitting back in a lab. So to really get why this one machine is so incredibly important, we first have to ask, why would you even want to use a biological nose to begin with? Well, the answer is actually pretty simple. Nature is just way, way better at this than we are. I mean, the difference is just staggering. Our best electronic noses are okay, but their sensitivity is pretty limited. A mammalian nose, on the other hand, can detect things down to the parts per trillion level. And it doesn't just give you a simple yes or no. It creates this rich, complex, high-dimensional pattern for every single smell. It's like comparing a stick figure drawing to a high-resolution photograph. There's just no contest. The powerhouse behind all this is the olfactory bulb. And you really shouldn't think of it as just a sensor. It's basically a tiny, sophisticated computer. You have millions of these specialized neurons that create a complex map of the smell. And it's this incredibly rich pattern that gives it such amazing power to tell the difference between thousands of scents.
Okay, so the idea seems brilliant, right? Let's just use one of nature's super sensors. But this is where you run into a massive engineering headache, a fundamental problem that you just don't have when you're working with silicon chips. And that problem, in one word, is variability. You see, unlike mass-produced electronics, where every single unit is a nearly identical clone, in biology, every single sensor is different. It's because, as the research says, no two olfactory bulbs are identical. They all have slight differences in size, neuron density, all sorts of things. And that means each one has its own unique personality, its own special way of responding to the exact same smell. So you can see the issue this creates for a network, right? If you've got three sensors out in the field and one reports a signal strength of X and another reports Y, what does that even mean? You can't compare them. You don't know if one is closer to the source or if it's just a more sensitive nose. It's like they're all speaking a different language. How do you get them to agree on anything?
And that is where our hero machine comes into the picture. The entire ODAR concept is only possible because of a single piece of hardware that was designed to solve this exact problem. It's called the ODAR testing chamber. Its purpose is singular and absolutely critical. It's a calibration device. Think of it like a translator. It takes one unique olfactory bulb, puts it in a perfectly controlled environment, and then it systematically maps out all of its specific responses. It literally learns that sensor's unique language. And calling it the gold standard is key here. This chamber isn't just another part of the system. It is the source of truth for the entire network. The accuracy of every single sensor out in the field is completely dependent on the precision of this one machine back in the lab. So how does it work? Well, let's peek under the hood. It's essentially a super sophisticated life support system, and its whole job is to keep that piece of brain tissue alive and functioning perfectly during the tests. And it all boils down to four core subsystems. First up, you've got environmental control, which manages the entire atmosphere inside the chamber. Then there's biosustainment, which is the dedicated life support, keeping the tissue healthy. Third is data acquisition. That's the electronic side of things, how the machine actually reads the brain's signals. And finally, you have analyte delivery, which is the incredibly precise system for injecting the scents for testing. The level of control here is just incredible. It uses something called a Peltier device to keep the temperature at a perfect 37.0 degrees Celsius. An ultrasonic humidifier keeps the air at over 95% humidity so the tissue can't dry out. You've got mass flow controllers managing every molecule of gas and a tiny pump delivering a constant, gentle flow of nutrients. It's basically a portable, automated biology lab in a box. Okay, so with the bulb alive and stable inside this perfect little environment, the chamber can finally start the calibration process. This is where it learns the bulb's unique language and creates what the researchers call a neural fingerprint. The whole process is very methodical. First, you let the bulb stabilize and get used to its new home. Then, you record its baseline activity when there's nothing but clean air. Next, you inject a very precise concentration of a chemical, let's say 50 parts per billion of toluene, and you record the response. Finally, you purge the whole chamber with pure gas before you start the next trial. By repeating this over and over with different chemicals and at all sorts of concentrations, the system builds a comprehensive profile for that one sensor. It creates what's called a dose-response curve that maps a sense concentration to that specific bulb's unique neural pattern. And this, right here, is the key. This becomes the labeled training data for the AI. You're literally teaching the machine learning model what, say, 50 parts per billion of toluene looks like to this specific nose. And this is where it all comes together. All that painstaking work that happens inside this one special chamber is what enables an entire network of sensors to function as a single, cohesive unit. It all starts with just one highly precise, lab-grade testing chamber. This is the master calibrator. But from that one chamber, you can generate the unique calibration files, the neural fingerprints, for dozens of individual field sensors. Each file is like a personalized translator that gets loaded onto the processor of the device carrying that specific bulb. So now when those sensors are out in the field and they detect something, they don't just send back their raw, confusing neural signals. Nope. They use their unique calibration files to translate the reading into a real, understandable number. So Node A can confidently report, I'm seeing 120 parts per billion, while Node B reports 95. They're all speaking the same language now. The network can just compare these apples-to-apples values and calculate a vector pointing right up the concentration gradient, straight to the source. It's brilliant. So you see, this one gold standard chamber is the key that unlocks the whole thing. It creates a personalized translator for every single biological nose. And this leads to a pretty fascinating question. Now that we have a blueprint for giving machines a near-perfect sense of smell, what should we teach them to find next? Medical diagnostics, hidden explosives, environmental threats? The possibilities, they're truly extraordinary.